Hello everyone, this is Dr. Ashank and I would like to speak about a brief update on coronavirus. This presentation and the following presentations on coronavirus and COVID-19 will be discussed in very briefly regarding various aspects and most important focusing on dental practitioners, dentists and dental students. As you all know, the current pandemic of COVID-19, which is caused by the coronavirus, is causing disasters globally and it is considered as a biological war in today's world. Coronaviruses are nothing but a group of viruses which are related to RNA category. So they are basically RNA viruses and they cause diseases in mammals and birds. These coronaviruses constitute the subfamily Orthocoronavirinae. That is the most important subfamily, which is Orthocoronavirinae. The etymology. The etymology means how the terminology or the name is derived. It is derived from a Latin word corona. Corona means a crown. The name refers to the characteristic appearance of the virion. Virion, as you all know, is the most infective part of a virus. And you see a bulbous surface, a huge surface with lot of projections surrounding the surface, creating an image which resembles or reminiscent of a solar corona. And this morphology is all created by the viral papillomas which you see on the surface of the virus. So these viruses are roughly spherical, bulbous surface with projections, and the average diameter of the virus particle is 125 nanometers with the diameter of an envelope which is 85 nanometers and the spikes are almost 20 nanometers long. And these are envelope viruses and they are single-stranded RNA genomic viruses with a nucleocapsid of helical symmetry. As we all know that a virus contains or the virus structure in general also has got a nucleocapsid. It contains a DNA or an RNA which is the inside part of the virus. Then there is a nucleocapsid surrounded by some viruses have got papillomas, which is basically the tegment wall of a virus. So this is wrapped in an isohedral protein shell and these viruses genome size ranges from 26 to 32 kilobases. So we can very clearly see in this pictorial representation of a 3D diagram of a coronavirus that the virus has got spike-like proteins which is basically a glycoprotein S. It has got again two subunits S1, S2 and then you have an envelope. Then inside you have an RNA and RNA protein. The coronaviruses are classified into alpha, beta, gamma and delta coronaviruses. And the most important point to be noticed is the alpha and beta coronaviruses, which are called as SCOV, are able to infect mammals, whereas gamma and delta coronaviruses are responsible for infecting birds. Now, infections in humans. In December 2019, there was a cluster of pneumonia cases which was caused by beta coronavirus, which occurred in Wuhan, China, which you all know and very familiar with. This coronavirus was initially termed as 2019 novel coronavirus, which is 2019 nCoV, and on 12 January 2020 by WHO it was given this name. WHO officially named the disease as a corona disease of 2019. That is what is an expansion and which is abbreviated as COVID-19. And coronavirus study group of the international committee proposed a name of a new coronavirus called as SARS-CoV-2. SARS-CoV-2. You need to understand SARS is severe acute respiratory distress syndrome. CoV-2, coronavirus 2, both issued on 11th February 2020. The Chinese scientists rapidly isolated the SARS-CoV-2 from a patient within a short period of time and then they were studying on the genome of the coronavirus. The studies estimated that the basic reproduction number for this virus SARS-CoV-2 is 2.2, which lies between an average or spans from an average of 1.4 to 6.5. I just wanted you to understand that basic reproduction number, which is called as R naught or R O, or reproduction number, is actually the number which represents that how many cases a single infected host can transmit to, but that is calculated to the susceptible host. Mind you that this number, whatever number, the RO number which belongs to a particular virus, is not a biological constant to that virus forever. Biological constant is something which is fixed to the virus. It is not fixed to the virus. This particular virus causes 2.2. That means it also depends upon the individual's immunity, the individual's reaction to the virus, the environmental factors, the comorbidities and all. So it is not a biological fixed value to SARS-CoV-2. That is why it ranges from 1.4 to 6.5 based upon the susceptibility and vulnerability of the patients, of the environment and of the geographic location. Previously, there were 6 coronaviruses which were identified as human susceptible viruses, among which we had alpha categories and beta categories. You can see the genomic number there. They had a very low pathogenicity and caused a very mild respiratory flu-like syndromes, especially just common flu like common cold-like. The other two beta coronaviruses were SARS-CoV and MERS-CoV. SARS-CoV and MERS-CoV, that is what I'm saying. Severe acute respiratory syndrome coronavirus and Middle East respiratory syndrome coronavirus. These two also had exploded in 2002 and 2003 when SARS-CoV-1, you can say, was first time had an epidemic in 2002-2003, and 2012 we had Middle East respiratory syndrome coronavirus epidemic. It was an epidemic, and the present situation 19 to 20 has become a pandemic, and these two were also fatal severe respiratory diseases. That means among the category of the 4 coronaviruses, alpha and beta were infecting humans. Among alpha and beta, 2 alpha and 2 beta were still causing very mild respiratory symptoms or common flu symptoms. Among the 6, 2 were very severe, which were called as SARS-CoV and MERS-CoV. It was found that the present genome sequence of the virus, which is called as SARS-CoV-2, is 96.2% identical to the bat coronavirus, and you can see the genome of a bat. But it had 79.5% identity or similarity to SARS-CoV-1, and MERS-CoV has 50%. That means it had an identical similarity of its genome sequence from the two highly virulent coronavirus genomic species, but then it was more matching with the bat. So based upon the virus genome sequencing results, it was suspected that bat could be a natural host of the origin of this virus, and SARS-CoV-2 might be transmitted from bats via an unknown intermediate host to infect humans, which we are still exploring. So it's very clear now that SARS-CoV-2 could use angiotensin converting enzyme 2, ACE2, the same receptor as SARS-CoV, to infect humans. So we need to also understand and focus that ACE2 receptor is the receptor which is absolutely susceptible to this virus, and the virus attacks or enters into the body or replicates through its reaction through ACE2 receptor. The ACE2 receptor is found in the lower respiratory tract of humans as a cell receptor for this virus, regulates both the cross-species human-to-human transmission, and this was isolated from the bronchoalveolar lavages of the COVID-19 patient. And then it was confirmed that this cellular entry receptor ACE2 SARS-CoV was confirmed by Zhao et al. The virion glycoprotein, which I said you, has got an attachment through its two subunits S1 and S2 to the ACE2 receptor. Now, because of the binding of this SARS-CoV-2 spike glycoprotein and this ACE2 receptor, this both unity or this both fusion is considered as a critical step for the virus to enter into the body and its replication, and that is why all the researchers are intensively focused on how do we have to inhibit this affinity or this reaction between the virus and ACE2 receptor. An emerging acute respiratory infectious disease, which is COVID-19, primarily spreads through respiratory tract by droplets, respiratory secretions, and direct contact or indirect contact. Now, otherwise, it was also reported that there were some SARS-CoV samples or specimens isolated from the fecal swabs of pneumonia patients, also on 10th of February. There was a group of authors, Zhang et al., who found that these were present in the fecal swabs and blood, indicating possibility of multi-route transmission. And ACE2 is present not only in abundance in lung alveolar epithelial cells, also in the enterocytes of small intestine, and also the recent research also says that ACE2 receptors are present in the oral cavity, in the salivary glands, on the tongue surface, on the gingival surface, and the mucosal surface of the oral cavity, which I will be dealing with the next presentation, the next chronicle presentation of coronavirus or COVID disease. Thank you so much.